lots of microglia from this math, and we stain it for an antibody for microglia, and we ask, are there more microglia in the part of the brain that has the high spiking? And the answer is yes. You can see these little dots here, and this is a blow-up. These are abundant in the high spiking, but not in the low spiking. So this is not something we would have predicted a priori, but it just jumps right out at you. And you can, based on the gene expression, you can say this is going to have more microglia, this is going to have 50% and 20%, and you stain it, and that's exactly what you find. Blood vessels. So you expect to see more blood vessels. You stain the tissue, and lo and behold, there's more blood vessels in high spiking than low spiking. This is the interesting cluster here that represents about half of the genes that form clusters. This one cluster of genes called neuron type 1 that's downregulated is 280 genes. Again, with 95% confidence, a really, really tight cluster. You can see by the cytoscape how tight that cluster is. One of the genes in that cluster is called ROR-B. We know from the literature that ROR-B is a layer 4 specific gene. Remember, we have 6 layers in the neocortex, so this is a layer 4 gene. We also found another gene called NUN in this cluster, which is a marker for neurons that we know we have very nice antibodies and we could stain tissue with. So the prediction is, if we get a piece of tissue and find a part that's spiking, and we stain it with an antibody to the NUN protein, we're going to see some dropout of neurons in layer 4. Does this all make sense how we do this? It's all kind of logical. The other thing we can predict, and I thought this data was amazing when Fabienne showed it to me, that wherever this population of microglia goes up, these neurons go down to the same extent. So where you see that dropout of layer 4 neurons, we're going to see microglia. And I thought he literally took the data of averaging 280 genes here and 200 genes here and just absolutely flipped it. But he didn't. So this has actually just happened to come across in this amazing inverse pattern. So what did we see when we went to do our validation study? We called this a microlesion. We had to make up a name for it because we didn't know what it was. So this is a normal staining using this antibody NUN. This is an upside down cortex, layers 1, 2, 3, 4, 5, 6. This is the cortical surface here. There's like 2 pieces of brain adjacent to each other. And this is abnormal. You can see you've got the neurons in little spots here, but this big hole right here at layer 4 as predicted. So we were showing this to our neuropathologist and saying, look, there's no neurons, just as we predicted. And he said, wrong, there's neurons there. What? There's no neurons. He was looking at the regular histologic stain called H&E. And if you look at that, it's perfectly normal looking under the microscope. So the standard histological method that a pathologist would look at would call this absolutely normal tissue. Because of this tissue biomarker we have, we've got this big hole. And if you use light microscopy, you see these little triangles here, those are neurons. So the neurons are actually not lost at all. There is no loss of neurons, but they're not happy. There's something wrong. So you take the next section over and the next section over and you stain with other markers to try to understand what's wrong with this part of the brain. This is a protein called MAP2. It stains the dendrites. Remember, neurons have axons that send things out and dendrites that receive input in. This big pale area shows there's no dendrites. So all the wiring connectivity in those deep layers is gone, even though the neurons are still there. And what else would you expect to find there based on the math? It's full of microglia. So CD68, just as you expect, when those neurons go through the change and no longer turn on those proteins anymore, we see the microglia. If you look using another staining method, so here's your normal area, your microlesion, and you stain it with this what's called silver stain, you get these beautiful columns of axons and dendrites in the normal tissue. They're gone in the microlesion. So there's definitely a wiring problem that we discovered, and we knew exactly where to look at with these tissue biomarkers, and now we're trying to understand what this means. Microlesions we found come in 3 varieties, either superficial, centered at layer 4, or 2 through 4. And when you go back to the clinical data, the more microlesions you have in a piece of tissue, the higher the frequency of spiking. So full circle, you come back to the electrical activity in vivo that we recorded in those patients' brains, and the more of these lesions we have in these deep layers, the more spiking you see. Finally, here's an example of a couple of microlesions where you actually have relative preservation of layer 2, 3. Remember we talked about layer 2, 3 and the low-hanging fruit of MAP kinase? Well, they actually have something to do with each other. So if you look at the next section over and you stain with your Krebs and your ERK antibodies, they go together. So putting it all together, what we think is, and again, it's a completely new discovery that we didn't hypothesize. We just used statistics and math to kind of help us solve this problem, that there's some microlesion or loss of inhibition that creates synchrony in the superficial neurons, and that's what causes an epileptic spike. So this is our working model right now that we're trying to understand and also develop therapeutics for. So to summarize this part, MAP kinase, CREB, superficial layers, seems to be associated with the spiking, and actually when we look in those areas superficially, they have tons more synapses I didn't show you. The microlesions are in the deeper layers, maybe lacking inhibition. Normally inhibition goes to the surface. Without that inhibition, you have synchrony. So trying to piece the story together. Question, is this required? So this is kind of interesting. It's esoteric. Maybe we understand something about epilepsy in the brain, but is this something we can go after and treat? Because we're physicians and we want to do something for our patients. Right now we're very limited what we can do in terms of drugs. So is this pathway a therapeutic target? So how do we go about figuring that out? We can't do this in humans. There's amazing what we've done in humans so far, and the reason we could do it is because it's part of their standard of care and surgery for their epilepsy. But we can't go in and knock out genes and pathways in our patients. Generally, they wouldn't appreciate that, and nor would our IRB. Actually, IRB doesn't appreciate much, but I think it's getting better, right? Okay, so we need to do animal studies and really try to find an animal model that parallels. We fail miserably in translational medicine in curing a mouse or curing a rat, whether it's an Alzheimer's, a cancer, or an ALS mouse or rat, and then we try to translate that treatment to humans and we fail miserably over and over again. So the goal was to say we have this huge number of biomarkers, targets, and genes. Can we actually find an animal model that looks like humans so that if we cure the animal, maybe it'll actually help people? So this is what we did, and this is the model we came up with. We use a tetanus toxin injection into the somatosensory cortex of a rat. When you do that, it transiently blocks or blocks GABA for a while, which is the inhibitory neurotransmitter, and develops spiking. And then we record both on the same side of the brain in 3 different locations on the opposite side. So the only injury to the brain is really that needle stick to put the toxin in. Everything else is outside of the brain. Here you can see here's our rat, and he's got his little cap on, which we can plug him into our amplifiers and record his brain waves. And so here is we have an 8-bed epilepsy monitoring unit for rats over in the College of Medicine Research Building. At different time points, we can look at the brain and ask, are these tissue biomarkers turned on? Are they in the same layers, the same pathways involved? So here's an example of a spike that's occurring at this site on the electrical recording. You can see over about 3 weeks, the spikes increase in frequency with the tetanus toxin versus the vehicle injection. The other thing we can do in the model, which we can't do in humans, is we can look at the time course of the development of the disease. So the spikes actually started very small and interestingly enlarge. So the field size, the number or the populations of neurons involved, is growing over this 3-week period. So this allows us mechanistically to start thinking about and looking, what is that connectivity? Do we see that same layer-specific connectivity abnormality that we've described in humans? And this is kind of a picture showing about the distribution of the size of the brain. And again, this is a rat brain, which is like the size of my thumb. When we're talking about human brains in 10 square centimeters, that's a lot more neurons that's firing at the same time. So one question is this a good model? Do we see the same things that we see in human tissue? This is the rat brain in coronal section. This is our stain for CREB. Remember, this is the layer 2-3 specific phosphorylated protein we saw in humans. And you can see on the spiking side versus the other side, beautiful activation of CREB in layers 2 and 3, just as we saw in the human tissue. So even though it's a rat, this part of the epileptic mechanism seems to be shared with humans, so maybe is something that we can go after therapeutically. So what kind of drugs should we use? How do we pick a drug to treat these rats so we can then maybe go back to humans in clinical trials? So we talked a little bit about the many different things that can cause epilepsy, from head trauma to tumors to strokes. Often it takes 6 months, a year, or longer after a head injury to have your first recurrent epileptic seizure. So something is happening over a long period of time, and we think it's this synaptic reorganization, these changes in networks that are occurring in the brain. So these cellular molecular responses lead to network changes, lead to synchrony and spontaneous seizures. Now, current drugs that we have for epilepsy now, they're really not epilepsy drugs, they're anticonvulsants. They prevent seizures, and as long as you take this drug every day for the rest of your life, if you're lucky and you get good control, you don't have seizures. Actually, a third of patients never achieve 100% control of seizures. So we need better drugs. So we're after drugs that actually can be preventive or could reverse or disease modify. If you take this drug, it prevents you from getting epilepsy, and maybe by looking at these potential pathways here, we can find an anti-epileptogenic target as opposed to an anticonvulsant. So what are some targets? Well, we have this data. We have our low-hanging fruit from the human brain tissue that can give us ideas. One of them, we talked about the MAP kinase, CREB. Of course, it's turned on in rats, it's turned on humans. Let's knock it out and see what happens. Plasticity genes. Inflammation, we saw these microglia in the human brains. And are these responsible? What if we have drugs? There's a lot of anti-inflammatory drugs being developed for degenerative disease like Alzheimer's. Neovascularization, maybe those blood vessels are contributing to the epilepsy, so if we can knock out the development of those blood vessels, that would do something. So these are just 3 possibilities based on the human tissue studies. Where we started again was our low-hanging fruit with the MAP kinase pathway. The nice thing about this pathway is it's been very well studied in the cancer field. There's a whole host of drugs that have gone to human clinical trials, so repurposing one of those drugs might be a quick way to get something to a patient with epilepsy and see if it has an effect. So we picked a drug in this pathway. It's called a MEK inhibitor. This is our target in the human tissue. This activated diphospho-ERK is, so if we block this, it should block that from coming, and that in turn should block the development of CREB activation. So we tested this in our animal model. So these animals did not get the drug, they got the vehicle, and they have CREB activation in layers 2 and 3, more on the left side, which was spiking more. Those animals that got this drug, SL327, within a week, they didn't activate CREB. So it actually hit the target in those animals. Next question is, well, what effect does that have on developing spikes in epilepsy? So here's our group of animals that over 3 weeks developed spikes. This is our sham injected with saline. They don't develop spikes. We took a third group of animals and gave them the drug just for 1 week after the injection of tetanus toxin and asked, do they develop spikes the same way as the control animals or not? And the answer is it prevented it. So this green line shows that the animals don't develop spiking, and even after you stop the drug, during this critical window, they don't develop the disease. So this is kind of exciting. I mean, this is kind of a morning after pill approach. So you hit your head, you take this drug for a week. There's a lot of challenges with drug development. There's a plasticity pathway. It might make you stupid by also taking this drug because that's an important part of the brain. But being stupid in the intensive care unit may not be a bad thing for a week after you get your head injury. And right now we're actually working with the UI Center trying to develop a small molecule that gets into the brain well so we can actually test this in first animals, then humans. So we need to design a clinical trial. We've got a target, we've got a drug, we've got some ideas. How do we get this to patients? This goes back to this idea of biomarkers. I talked about tissue biomarkers of things in the tissue, but a clinically useful biomarkers is something you can use to say that this group of patients is more likely to get the disease of epilepsy than someone else. So when you design your clinical trial, if you know that everybody in that group is going to get epilepsy, it keeps your N down, it keeps the cost down, so pharmaceutical companies are actually interested in supporting the trials. So how do we find a biomarker? The gold standard we have right now really is this electrocorticography. It's taking the skull off, putting electrodes on the surface of the brain, and we can see with beautiful precision all these different spike and things long before you develop seizures in the brain. And this is a study done by John Ebersol, University of Chicago, with James Towle. This is a routine EEG. So this is an EEG electrical recording from a scalp recording. You can see a couple of sharp waves here. If you look at that same segment of recording with the electrical grids, you can see the resolution is just hugely better. You see these two spikes, but you see the brain spiking all the time. So this is not sufficient because by the time you get this point, you're too late. So how can you get this resolution of seeing which part of the brains are epileptic and if somebody's going to develop epilepsy or not so we can design clinical trials? So we need non-invasive MRI biomarkers. Non-invasive, in other words, not taking somebody's skull off or doing PCR or taking out pieces of brain to see if somebody has epilepsy. So what we've talked about is mostly RNA studies where we look at gene expression, and that's very, very helpful to find these cellular and tissue mechanistic biomarkers. But how do we get a non-invasive biomarkers? Well, metabolomics or MRI spectroscopy is a way of seeing many small molecules in the brain non-invasively, and we can do this both ex vivo on tissue that comes out, and we can also do it in vivo using a routine MRI magnet that our patients go in all the time. So maybe by taking these samples and doing metabolomics, we might be able to get a non-invasive biomarker. So this is the metabolomic profile of human neocortex. These are the various different metabolites, GABA, glutamate, lactate, things that you can resolve at very high spatial resolution. This goes in 11.7 Tesla magnet, as opposed to a regular human magnet is 1.5 or 3 Tesla most of the time. So just asking the question, if we take paired samples again between a high spiking and a low spiking area, And doing the same study design, do we see a consistent difference in our Venn diagrams between 9, 10, 12 patients? And we did. We found certain metabolites. Oh, I'm sorry, this is an old slide. We published this, so before we published it, I had to call them metabolite 1, 2, 3, 4, and sent the patent off and stuff. So we found metabolites that are very predictive of the epileptic areas. And this is work by Helen Wu, MD PhD student. You can see, using this what's called a logistic regression model, highly predictive with good sensitivity and specificity. Give me a piece of tissue, I can tell whether it's epileptic or not using the metabolomic profile. So that's really exciting because that's something that potentially could be translated to a non-invasive method. So next step is how we turn the invasive tissue biomarker into a non-invasive method that we can actually use in patients. And for this, we have a three-pronged project that Helen was orchestrating, and we're continuing here. The human ex vivo data I told you about. We have human in vivo data, and we have rat in vivo data at different strength magnets. And together with Xiaolin Yang in biomedical engineering and Xin Cheng, who's an MD PhD student, we're starting to go over some data sets where we have patients who have epilepsy in focal areas. We have intracranial electrical recordings. We can now start building the metabolomic profiles in three-dimensional space, building the electrical profiles in three-dimensional space, and developing AI programs and spatial programs to really hone in on those areas that are predictive to be epileptic. And if this works,